In this lecture for my Bioinformatics class, Bio for 510 at California State University, Monterey Bay, I'm going to be talking about some of the stats that are behind the BLAST algorithm and the probability distributions that we're applying to it. To get a better understanding of a good case how we take sort of a non-standard approach in bioinformatics as opposed to sort of what we might think of as sort of a Gaussian standard parametric approach to statistics. And I think it's informative to see sort of the different ways that we apply these things and these types of techniques in sequence alignment to better understand the kind of flexibility and possibilities that are out there for us. So hopefully by the end of this class, you'll have a better understanding of how probability distributions are applied and used as evidence, and specifically how the extreme value distribution can be applied to a data sample, and also be able to understand how to calculate and interpret BLAST e-values, which are generated by this extreme value distribution, probability distribution. So, last class we talked a little bit about local and global alignments. Global alignments are when we know our boundaries of our queries, and generally we think indels are important. We use them a lot when looking between species. Local alignments are where we don't really know the boundaries of our query, where we're not really concerned about indels, and are particularly useful when trying to find conserved sequences or identify sequences either homologous or functionally convergent because of these shared characters. We had our classic BLAST result that we talked about last class, right? And here's an example of something you might get back. And there are some components in there, when we think about like what makes up a result, right? Notice that you're getting back, an identified object that is this hit that you're getting back. We can identify some other entities of this, right? So you're going to get back an alignment of the query, the thing you were searching, and the subject, the hit that you got back from the database. And then you're going to get some other information. And we talked a little bit last class about how, we calculate some of these similarity scores. And notice that, we have our number of identities, our number of positives, our gaps, right? All these things will go in to calculate our similarity score. But you will often get back both a expected value and a bit score. And so what are these expected values and bit scores? A lot of times people look at those expected values and interpret them as being p-values, but they're not. They're not p-values. They're not even on the same scale as p-values because unlike a probability value, expected values can actually go above one. So what are they and how are they calculated? And how can these entities be related to our confidence that our matches in BLAST are not just made at random? So just to remind us, the BLAST algorithm, we had this sort of set of steps that we were walking through, and at the end of this step, we would have identified this high scoring segment, right? This HSP, so this high scoring pair. And part of what we're going to try to do there is evaluate the significance of that pair, right? Well, what does that mean, evaluate the significance of that pair, and how are we going about doing it? We talked about, that the alignment part of it, and so the alignments where we're actually taking all these words and matching up and finding these high-scoring words, right, and extending those things to make these HSPs, these high-scoring segment pairs. But how are we actually evaluating that significance? We haven't really talked about that. And especially because when it comes down to it, it's these scoring patterns and this threshold E value, which is actually going to determine what's going to be reported. If we had to report everything, it would be an enormous amount of information, right? So we do need to apply some sort of way of filtering things out. And also giving us confidence in what's a better alignment versus another alignment. So to do that, we're going to use a distribution that's known as the extreme value distribution. And I don't know if any of you are all that familiar with it, but it's a different type of distribution. So it's a distribution of a random variable that is itself the maximum of a set of random variables. It's one that's been widely used in bioinformatics to assess whether an alignment score indicates biological significance of an alignment. And an example that we'll talk about a lot in this class or this lecture will be the if you have the height of women following a normal distribution, which you can see down here in blue, right? The maximum height, or y, of a randomly selected group of these women in blue is actually also random. So I select 10 women, and the tallest woman is 6'2". I select another 10 women, and the tallest woman is 5'8". I select 10 women at random from this population of college women, and the tallest woman is 5'11". You get the picture, right? And so as I pull in these maximum tallest women, so say I have do a sample of 25, I end up with a different distribution than this distribution of this density for college women, right? So this is the height of all women. This is the distribution that if I sample 25 of all of these women, that I'm going to get for what their heights are. So the tallest woman of a group of 25. It's a little bit weird, but I hope you get to see that there is this thing. So this is a distribution of the maximum height of any woman of a sample of 25 from this larger group. So you might be like, okay, great, like you get this distribution, but so what? So let's review a little bit about what statistics, how it functions and how it works. So if we think of the extreme value distribution, right, we often define distributions using something called a probability distribution function. And I've listed this equation here, right? And what we can tell from this equation, right, this is going to what's make up the curve of that distribution. This is what the function that makes up this red curve here, right? That's the probability distribution function. And you can think of it as, what is the probability of a specific value? What is the probability of -1? What is the probability of -2? Or in this case, what is the probability of 70? What is the probability of 71? What is the probability of 72? And that's what this function will help us calculate, right? And we can derive a mean and a variance from this function just like we would for a normal function or a Gaussian function, right? But the equation is going to be a little bit different, right? It's because the parameters are different. Notice that we've got this k hat and this lambda parameter that are kind of driving the dynamics of this equation. In addition to the probability density function, you'll often hear people talk about a cumulative distribution function, right? And the cumulative distribution function looks something like this. And what this is this is the probability that we would get for the probability of a value being greater than or less than a specific value, or probably being of a value pulled at random being less than or equal to a specific right? And so it's this cumulative distribution. So we're either looking at one side or the other side, right? And so this is why cumulative distribution functions are typically plotted as a sort of an S-shaped curve, because as we get up to a saturation point. So it's important to realize that these functions are underlying all of the distributions that we use. And so the functions can change dramatically depending upon what kind of distribution we're using as our underlying distribution. But what they're going to do is they're going to approximate the distribution of any random variable. And in this case, what we're actually approximating is the distribution of the random variable that is the maximum value of a large random sample, right? And if we are trying to define the distribution of any random variable that is the maximum of a large random sample, in other words, what's the tallest woman of a group of 25, for instance, not that large of a sample, but you get the picture, it's going to be defined by two parameters, right? In this case, that's this lambda and this k hat. So just like any sort of distribution, when we go out and test it, right, we're going to be able to calculate a mean and a standard deviation. And we would do that how we would normally calculate a mean and a standard deviation. But interestingly enough, we can also calculate these values of lambda and k hat based upon the mean and the standard deviation. So you can see that lambda tends to is closely is defined basically by the values that we get by the standard deviation, so it has to do with the variance of the distribution, whereas k hat includes both of those factors. So this is more driven by sort of the mean behavior of that distribution. One thing to note is that when we use the extreme value distribution, what it does is that we're actually using larger sample sizes and larger groups to give a closer approximation of the extreme value distribution of a situation. So if we looked at the actual, in this case, like the blue line shows what the actual density function is, right? And this is the density function for when we sample 1,000 women. So you notice versus 25 instead of 1,000. At 1,000, we see something that closely matches what the actual mathematical function would be for this. If we're trying to get to values of these lambda and k hats that are as close as possible to these two, we'll often use a method called maximum likelihood, which is our way of estimating these parameters. So what's the big deal with this distribution? Well, just to get back to sort of like how we might think about things. What I think it's informative to think about this issue in relation to the idea of a right tail p-value. So if we think about a right tail p-value in traditional statistics, right? So we know what a p-value is. The probability we're talking about here is the probability of seeing a value of a random variable that is larger or as large as a given value. So what's the probability, in other words, that if I pull a value at random, that I'm going to end up in this tail, that I'm going to get a value that's in this tail. And if I think about this, right, basically this is that cumulative function. So this is, if I think about what's the probability of x here, right, what I'm actually trying to figure out is, well, what's the probability that I'm going to get a random value that's greater than x? So here we can go back to what we did previously, where we wanted to calculate, what is greater than a specific value by calculating what is less than another specific value. In other words, the complement of that probability and subtracting that from 1. If we think about, what this means in terms of these cumulative distributions, it just means that if I'm going to calculate the probability of x being greater than a value, all it is just 1 minus the cumulative probability distribution. And for a normal distribution, that would look like this. For an extreme value distribution, since this is the cumulative distribution function, this is how I calculate that value. So this is how I calculate the probability if I pull a random value that I would be in this right tail. I think it's important for us to remember what we're doing. So I've sort of talked about it a little bit for probability, that when we think about how we're what we're actually doing when we're doing these probability calculations, we're actually, if we go back to our discussions about continuous random variables that I brought up earlier, what we're looking for is, the probability that a random variable value lies in a specific continuous interval or collection of intervals. And that's what that probability density function is doing, right? So that probability density function, right, is letting me figure out, what's the probability of getting a value in C, what's the probability value getting in D, all this information, right? And I can add up all the probability values that I get between this, right? And I can figure out effectively how much area is under this curve or is under the density function over a specific interval, right? And if I think about the entire curve, the entire area under this, because all probabilities have to add up to 1, right? All of the probabilities, the set of probabilities have to add up to 1. I know that all the area underneath this curve is actually equal to 1. Does this look like anything you remember from calculus, right? Like this is integration, right? Like this is how you would generate the area under the curve. And that's, we don't always think about statistics being tied to calculus, but it is, right? And we don't always think about calculus being applied, but whenever we're doing probability, we're actually applying probability theory, we're actually applying calculus. Not that we think about it that way. But so what we're looking at, and if I add up this area, right, this is the probability space, this is the set of outcomes that would be associated with getting a random variable between C and D. And, if we think about our normal distribution case, right, and we have our own probability distribution for a normal distribution, right? We've got and that would have a standard distribution look like this. One of the things that I can do, right, is we talked a little bit about this before, is if I had a different mean and variance for my distribution, I can still fit my density distribution to it by recalculating the values at each of these x values into z-scores. Right, so I can transform any normal random variable into this sort of standard normal distribution. And if I can do that, then it becomes really easy for me. I can apply that probability distribution function and start to calculate the areas under the curve fairly easily for each of these corrected values of x. And if we think about this from a data perspective, right? Like, so we might have some actual data that we would have collected. We can then scale our counts so that they're on basically everything is on that scale to a 0 to 1 scale. And then we can fit our probability distribution to that. So now the entire space underneath this distribution should be equal to 1. And then if we want to see, like, what's the probability that if I pull a value at random, that it's based upon this distribution, this probability distribution function, in other words, based upon this curve, what's the probability that I'm going to get a value that's sort of in this range, right? I can use that density function and that interval of x to calculate the area underneath that curve. And this is what I'm doing for looking at the probability of outcomes, right? And from that, right, I can figure out, well, what's the probability that I'm going to get a value between, say, 2 and 3.5, and the cool thing about the normal distribution, right, is that it changes based upon what the mean and the variance are of the distribution, but I can always calculate it back to this original probability distribution just by transforming the data, weighting the data by shifting the datas by the mean, and then compensating for the amount of variation or the spread of the distribution. So I can always transform this normal variable back to a random variable. And that's important when I think about other distributions as well, like the extreme value distribution, right? So keep that in mind. Like I can always fit a distribution to my data and then recorrect it to get it back into something that's a little bit more standard, right? And then you can do things like take the probability take the cumulative distribution function and use that to figure out, like, well, what's the probability I'm going to get a value less than less than 2, or what's the probability that I'm going to be out in this tail, right? And then now we can start to see how it's looking more and more like that p-value approach to assessing probability distributions. And if we want to think about we talk about confidence intervals, for instance, right? So we can measure the mean and the standard deviation of a distribution, and we can figure out, okay, I can use that original distribution, right, where I know that plus or minus 2 standard deviations is going to account for 95% of the variation, right? If I know that mean and variance, I can calculate these z-scores, go back to these to my original probability density function and my cumulative distribution function and calculate what's the probability that, based upon that mean and variance, what's the probability that I'm going to get a value between -2 and 2, right? And because I can use this z value, I can actually calculate like what does 2 actually mean in terms of the thing I really measured. So again, point is we're using these functions to calculate the area that's underneath this curve, right? And that when we think about distributions, that's how we want to think about them, right? We're using this to calculate the area underneath the curve. So again, we were talking about the extreme value distribution, right? We talked about this earlier. Just to remember, it's the distribution of random variables that is itself the maximum of a set of random variables. So in other words, we sample from some sort of population, say college women, and we sample 1,000 of them. What's the largest height of that group every time, right? And that has a very specific distribution. And here it's somewhere between, 72 to 76 plus inches. And this distribution, just like a normal distribution, has a mean and has a variance, right? And we can calculate that and convert that into these lambda and k hats that we can then use to estimate the probability distribution. So in this case, what we're really looking for here, right, is we're going to be looking at this tail. Like, even if we think about what we're looking at for large distributions, right, we're really looking at what's in the tail distribution as I sample randomly. So if we think about a specific case, again, this case of the tallest women of a population of women on a college campus, and again, what we're looking at in this extreme value distribution is the distribution of the maximum height if I choose a group of women at 25 to sample at random, right? What's the maximum height of that group? So if I sample 1,025 samples, this is the distribution of maximum heights that I get. So it's representative of this tail of this distribution, right? So if I could ask a question and say, well, based on 25 women randomly selected across 30 college campuses, what is the probability that if you sampled another college campus where the maximum height of a woman was 77, that height was not the height of the tallest woman on campus? So what's the probability that I'm going to get, if I sample 77, that I could find another woman who's actually taller? Right, and this could be a very useful thing to calculate, right? Because you might want to know, well, do I have to keep sampling to get this distribution, or can I stop? And so what you can do is you can say, well, I can look at this distribution of these 30 college campuses and 25 women and I can calculate the mean, and I can calculate the variance of that distribution. So I could have a mean of about 70 and a variance of 1.5. And if I look at these numbers, this is kind of the numbers you could use to calculate that. Based upon these, this mean and this variance, I can calculate my values for alpha and my values for k hat, right? And so this is just 1.252 divided by the standard deviation, right? So this is the variance, so I have to take the square root of that. And then I would calculate k hat, right? So I get my two values for these two entities, right? And then I can just plug that into my probability distribution. So what's the probability that I'm going to get a value that's greater than 77? And that's going to be equal 1 minus the probability that I'm going to get a value that's less than 77. And that can be calculated using this equation, right? So I can just plug in these values, right? And I can figure out, based upon this value of k hat and this probability of lambda, what's the probability that that this woman was not the tallest woman on campus, right? So what's the probability that if I had a woman who was 77 inches that she'd not be the tallest? And the probability is quite low, right? So there's a very low probability that I'm going to sample somebody who's going to be taller than 77 inches. So that's the idea. What's the probability that I'm going to get something even better? What's the probability that I'm going to get something even taller? What's the probability that I'm going to get something that scores even higher? That's the idea here, right? So when we think about sequence alignments, the goal is to quantify how likely a specific similarity score between two sequences would arise by chance. Is this just random? Like if I just pulled out a random value from this example, would I get a better score? What's the probability? If that probability is really low, right, then I might start to be able to make the connection that this similarity is significant in some way, right, and biologically meaning. And so historically, when we first started off with this, biologists were actually applying a normal distribution to this case, and they were using z-scores to evaluate biological significance. But what they found was that actually if we looked at these highest score values, they didn't follow a normal distribution. They followed an extreme value distribution. And I hope from the arguments I've made previously, you can see why. Because we're identifying what is the maximum or the highest score of a random search, right? So of a set of random variables. And so what it came to, that equation that we see is just slightly converted, right? So this k hat is now going to be weighted by the length of the query, so how long your sequence is, and the number of items that we might find in our database. So there's a little bit of a shift here in terms of what we're targeting, but we're still using this parameter lambda and we're still using this parameter k hat. But now we're testing whether or not S, or a similarity score, is, I could find a better, what's the probability I could find a better random variable that's better than the similarity score I calculated. So let's look at an example of a BLAST output. So if we have a human p53 alignment to the UniProt Swiss Prot database, and we only look at C. elegans, for instance, what's the effective length? And we have some information, like we know that the effective length of the query is 305 amino acids long. We know that the length of the database, the number of items in the database that we're querying is like 1.4 million. And from that, we can calculate. We also know a little bit about everything else. So with that information, we can calculate k hat. We can calculate lambda, right? And we have values for that. We know that we might have a similarity score, say 77 in this case. And our question is, if I have this similarity score, is that alignment biologically significant? Or is it just something that I could easily find something random that would replace it? So we're going to apply the same kind of equation here, right? We're going to have this value. We're going to try to see, well, what's the probability that I could get a similarity that's greater than or equal to 77, right? And I have this equation to calculate that using that cumulative distribution function. And so when you run the BLAST result, you get back something that looks like this, right? And the two things that I want you to key in here are, one, there's an expected value, and two, there's a bit score, right? I'll get into more detail about those bit scores, but basically a bit score is the measurement of similarity between sequences independent of the database size. But we do have this e-value, this expected value, right? And so what is that e-value? Well, when I the e-value is the calculation of this probability, right? So, sorry. So again, I have this case right now where BLAST reports that I've got this bit score, I've got this similarity score of 77, and I've got this e-value of 0.2. So I've got my similarity score, my bit score, and my e-value. And I want to know if this is biologically significant, right? So how do I calculate the right-tailed p-value for the data provided? So if I want to do that, I can just plug it into the previous equation that I have, right? Using this equation and the value of 77, right? I can go through and I can calculate that the probability is going to be about 0.0206, right? So that means that there's a low probability of me seeing a value equal to or larger than 77. How am I supposed to interpret that? I think what it would suggest is that we have a sequence here that we might assign to be one of a distant relative. Now that p-value is not exactly the same thing as the e-value, right? That 0.20 value. And that value is calculated slightly differently, right? So that value is calculated using that similarity score, right? And then we have E, which stands for the expected number of high-scoring segment pairs for each score of S. So in this case, for each score of 77. So that's what E is. The E value is actually the number of expected high-scoring segment pairs. It's not the probability of getting a value larger than a specific value. So it's the expected number of high-scoring segment pairs with a score of at least S, in this case 77, assuming random sequences. And you'll see that how you calculate that e-value, right? You're using a lot of things that we would have identified before, like k hat, and m, and n, and lambda. Right? And if we look at that, what we do is we notice that the e-value for 77 is pretty darn close to the p-value for 77. And because of these similarities, right, we also can see in this equation too, like if we look at this equation and we go back and we look at this equation, we can see that there are definite similarities between these p-values and the e-values. But it's not quite the same equation. And so we actually can calculate e-values from p-values and vice versa if we really want to. And the thing to notice is that when they are small, they're actually almost identical. But the reason why people tend to favor e-values is when they are large, e-values are a lot easier to work with than p-values. Because once we get to larger p-values above 0.05, basically they become a little bit difficult to interpret. Where the e-value is actually giving us a number, the number of expected high-scoring pairs that we might expect of at least that score, assuming random sequences. However, I hope it's clear that there's this strong connection between how we might use the extreme value distribution to calculate a p-value and how we can also use it in parts of it to estimate these e-values. And so it's a unique application and interpretation of this type of probability distribution. If we think about how we interpret e-values, basically any e-value greater than 0.1 we would think about as being insignificant. Anything that's 10 to the minus 5 to 10 to the minus 1 we would consider to be distant relatives. Anything that's between 10 to the minus 50 and 10 to the minus 10 we would see as closely related, if not homologously certain. And anything that's 10 to the minus 100 to 10 to the minus 50 we would identify as basically identical. And then sometimes you can just get even smaller than that, and it's basically an exact match. So that's the e-value, right? The number of expected high-scoring segment pairs that we might expect given the level of similarity. The bit score is a little bit different. The bit score is a base 2 logarithm of the size of a search space for of random data. So that's this m times n, right? And remember that if we go back and think about it, right, the n is the effective length of the database, and the m is the length of our query. So when we think about this bit score, right, it's actually estimating the size of the space that we would need, that would have to be scanned in order to get one high scoring segment pair of a similarity score S. Like, it's based on that idea, and it's a base 2 logarithm, so it's not the exact size of the thing, right? So it's condensed as a score to make it easier to look at. And so one of the things that this tells you is that this score does not depend on the size of the database that was actually searched, right? It's generating what you would need to, like, how large a theoretical search space you'd have to do just to get back a random example of this. So this is really useful because, like, a lot of times we might be comparing results from different search spaces, from different parameters, right? Maybe we're comparing searching one database versus another database. Is this a good hit? Are we always, sometimes, we might just be looking for a way to compare the results of different searches, and we want to find a way to do that. This is a way to do that, because what it's telling us is not telling us about the actual search we did, so the expected value given the search that we did. It's telling us about the search that we would have to do to get just one scoring hit at random. So when we think about it, if we have a score of 3.27, which we have in this case, right, the corresponding search space is going to be about 2.7 trillion, right? And the actual search space in this case was a lot less than that. The actual search space for this search was actually about 431,000. So that would tell us that what we found was not something that we just found at random, right? That would tell us that that's the level of confidence. Now, if I see something that has an even higher bit score, that means that it's an even better match, and that the search space that I would have to explore would be even larger than this, right? And that might mean that it's even less random that I saw this result. So that's what we're looking at when we're looking at these results, and I hope we'll practice a little bit. We'll give you a few examples on how to calculate these out by hand, but basically it's just about plugging into these equations. But I wanted you to see, like, what we're looking at. Remember, this is the number of expected high scoring segments given this level of that you would find at random, given this level of similarity, right? So and we have this other example of measuring sort of like what's the search space would have to be, this scoring mechanism. And I would say that, typically in practice, these expected values are particularly useful in ranking the results of a specific search, whereas the scores can be used to compare results from among different searches. All right. I hope that helps you understand some of the ways that we're applying statistics and probability distributions. And I think that this is a good example of how we take a distribution like the extreme value distribution, which is not one that you're probably normally used to using, and apply it in this very interesting case.